Hello, and in this podcast we're going to go through essentials around the gram-negative cell wall. So hopefully by the end of it you should be able to do the following things. So what we're going to do is we're going to briefly sketch some of the important things around what the gram-negative cell wall looks like and what it's made of. It's a bit more complicated than the gram-positive cell wall, but you should be able to identify where you find the inner membrane, the peptidoglycan layer, the outer membrane, and then one or two of the specific molecules and proteins found in the cell wall of a gram-negative bacterium. With the other podcasts, you should also be able to name some of the differences between the gram-negative and the gram-positive cell walls. Okay, so what we're going to do is we're going to start, as we did for the gram-positives, and we're going to begin by drawing an inner membrane. So the inner membrane between the gram-positive and the gram-negative cell wall is pretty much the same. It's a lipid bilayer. If you're a molecule, a water-soluble molecule, the only way in and out of the cell is to go through a transporter or a particular channel. There's no way that you can diffuse through the lipid bilayer. If you're a gas, however, you can. So things like oxygen can diffuse through, no problem. One of the main differences between the gram-positive and the gram-negative cell wall is the thickness of the peptidoglycan layer that's present. Both of them have it, but with the gram-negatives, it's really thin. So I'll just depict it as three little lines here. And as for the gram-positives, remember what happens is that the peptidoglycan is made up of alternating sugars. The sugars are synthesized inside the cell, they're exported out, and they're then built on the outside of the cell where the cell links them together. It's important to say, really important in fact, to say that the cell can not only build it, but it can repair it. And so there are repair mechanisms, there are repair proteins which are exported, and some of these repair processes are inhibited by some antibiotics that you can prescribe. Very quickly then, what we'll have is a thin layer of peptidoglycan, and as for the gram-positives, this layer is cross-linked, and that just provides the structural stability, which stops the cell from exploding. Another really important difference between the gram-positives and negatives is that on the outside of the cell, we have another lipid bilayer, and this is the outer membrane. What you have then is inside and the outside of the cell, and a space in between, which we call the periplasmic space. The periplasmic space is a really important site for protein folding, for antibiotic detoxification within gram-negatives. So it's not something that's there to be ignored. And hopefully, as I go through, its importance will become a little bit more clear. Now, I said that there are some specific molecules found in gram-negatives, and there's a molecule called LPS, which is called lipopolysaccharide. And LPS is found here, sticking out. This is just a cartoon version, so it's not entirely accurate. But what you've basically got is essentially an LPS layer on the outside of the cell. And that in itself can sometimes provide some protection against toxins, antibiotics, and so on for the cell itself. Now, let's imagine that you're a solute. How are you going to get into the cell? Well, you're going to get through the outer membrane by going through a type of protein channel called a porin. Porins are not really transporter proteins. What they are large trimeric protein complexes. A trimer is three proteins which link together, and this provides a large channel that allows molecules to enter the periplasmic space. They're not selective, or they're not that selective. However, let's imagine that we're charting the progress of an antibiotic. Let's say we draw a molecule here and we're charting the progress of penicillin. Okay, and penicillin works by attacking the peptidoglycan layer there. Penicillin's water soluble, so it can't get through this barrier here, so it's got to go through a porin molecule. So it enters the porin, enters, and then its mode of action, its site of action, is the penicillin, is the peptidoglycan layer that's there. Porins, though, are proteins, and that means the proteins are going to be synthesized inside the cell. They themselves are going to be exported into the periplasmic space, and they then track through and are then inserted into the outer membrane. And the cell has lots of different machineries which allow it to do this. But the fact that it's a protein means that it's encoded by a gene. The fact that it's encoded by a gene means that it can be mutated. And so porins can be mutated, which can sometimes preclude the movement of penicillin's antibiotics enter the cell. And if that happens, what that basically means is that penicillin is unable to find its site of action, which is the peptidoglycan layer. So that's one way in which gram-negatives can evolve resistance. And if you look at the kind of organisms for which there's a dearth of new antibiotics coming through, gram-negatives are nearly always at the top of that list, and that's because of the outer membrane, or largely because of the outer membrane. There's a few other reasons why as well. So let's imagine we're talking here about, this is this particular box here represents a protein export channel. What you can also have are enzymes can be exported as well, and they can go through and they can find their way into the periplasmic space. And some enzymes can be involved in antibiotic detoxification, so things like the beta-lactamase enzyme. So that's one way in which the cell can actually pump detoxification enzymes into this region. In a nutshell, there we have the outer membrane, there we have the cell wall of a gram-negative. The important things to note are lipopolysaccharide, the outer membrane, the thin peptidoglycan layer, the fact that we have an inner membrane, and lastly, the fact that we have a relatively thin cell wall. And the thickness differs from organism to organism, but it's about 5 nanometers to 50 nanometers. And it's a bit thinner than the gram-positive, which can vary from between 40 to 50 to 100 nanometers in thickness. Okay, there we have it. On to the next.